Growth of basil in simulated microgravity. Monday, March 12, 2018 to Monday, March 19, 2018. Plants use a variety of hormones to influence their growth through different processes, known as phototropism, growth by sunlight, thymiotropism, growth by touch, and gravitropism, growth by gravity. The following study focuses on gravitropism. In this process, the plant uses gravity to determine which direction its root and stem should grow. Problem: how will a plant grow when tilted 90 degrees? Hypothesis: if a plant is grown in a simulated microgravity environment, then the plant will grow straight outwards from the cup due to the elimination of gravitropism as a factor. Experimental design. In this study, there will be 3 groups, 1 control and 2 experimental. The control group will consist of normal growing conditions, where the seeds are planted in a cup with an opening at the top. The first experimental group will consist of cups tilted 90 degrees so that the opening is on the side of the soil. The last experimental group will use a homemade clinostat. A clinostat is a device that attempts to negate the effect of gravity on biological systems. It does this by placing a cup on its side, such as in the first experimental group, and then it rotates the cup. By continually rotating the cup, the plant will not be able to determine the direction of gravity due to the constant shifts of the direction of the force. The independent variable is the direction of gravity relative to the cup the seeds are planted in. This will be manipulated by changing the direction of the opening of the cup and the use of a clinostat. The dependent variable is the patterns in plant growth, which will mostly consist of qualitative observations of the stem and the roots. Constants include the type of plant, the cups, the light source, the elimination of supporting structures, the soil used, the amount of soil, the amount of seeds for each cup, the amount of water the plant receives, the time, and the place. There will be 3 trials. 3 cups will be in each group. Materials: basil seeds, plastic cups, soil, water, plastic wrap, a large box, duct tape, white Christmas lights, zip ties, cardboard, hot glue gun and hot glue sticks, small containers, motor/power drill, outlet, and metal rod. Procedure. Number 1. Using the duct tape, tape the Christmas lights throughout the inside of the large box. Ensure that the lights are evenly distributed through the box and that the plug is outside of the box. Ensure this is plugged in. This will eliminate phototropism as a possible confounding variable. Number 2. Using the cardboard and hot glue, create a small 5 cm by 5 cm by 10 cm box. Place this in the large box. This will act as a support structure for the first experimental group tilted at 90 degrees. Number 3. Using hot glue and duct tape, create the homemade clinostat using a drill, metal rod, cardboard, and small container. Create a cardboard disc with a diameter of 20 centimeters. Poke a hole in the center. Create a cardboard support structure by creating a 20 centimeter by a 10 centimeter by a 5 centimeter box. Place a small container in the large box. Place a drill on top of the container and duct tape it down. Place the disc on the metal rod and place the metal rod in the drill and tighten it. Place a support structure on the other end of the metal rod. Number 4. Add 5 milliliters of soil to each of the 9 cups. Place 10 to 15 basil seeds in each cup. Basil is an edible plant and grows relatively quick. Add another layer of dirt on the top of the seeds and give each cup 10 milliliters of water. Cover the cups with a plastic wrap. Number 5. Place 3 cups normally upright in the box. Place 3 cups on their side on top of the support structure. Attach 3 cups to the clinostat. Number 6. Plug in the drill and using a zip tie, tie the trigger so that the drill spins at about 1 to 4 revolutions per minute. Number 7. Close the top of the box and observe the cups daily for 1 week. Add water about every 4 days. Due to an error with the clinostat, 3 of the cups in the experiment were ruined. Thus, 6 total cups remained.
Most of the data taken was qualitative since the study focuses on stem and root curvature. In order to obtain this data accurately, an advanced computer program would have been the best choice. However, with the time frame provided, it was decided to focus on qualitative data. For the control group, there was a total of 30 basal plants that grew. As seen in these images, the plants grew upwards towards the sky for the most part. In one group, the plant stem seems to be leaning at a slight angle. The visible roots seem to grow downwards towards the force of gravity. For the plants grown in a 90-degree tilted cup, a total of 25 plants grew. As seen in these images, the plants grew out of the soil and immediately bent towards the sky, away from gravity. When viewing the side of the cup closest to the ground, many roots are visible. For the plants grown in simulated microgravity, 14 plants grew. Since the cup was often in rotation, much of the soil was moved around inside the cup. Much of the soil resided on the side of the cup as opposed to the bottom. As a result, many of the plants in the cup grew from the walls towards the center of the cup. There is a trend in the quantity of plants in each group. Plants grown under normal ideal gravitational conditions, such as in the control group, saw the highest quantity at 30 total. Plants grown in an environment tilted 90 degrees and under normal gravitational conditions saw the next highest at 25 plants. Lastly, plants that faced the stress of shifting gravity had the least growth at 14 plants, less than half of the control group. It is possible that the more a plant deviates from ideal Earth gravity, the less likely it is to grow. When looking at some individual plants from the control group, they appear to be mainly straight from the top of the stem to the bottom of the root. This aligns with the effects of gravitropism, which causes the stem to grow upward and the roots to grow downward. When comparing this to a plant from the first experimental group, or those tilted at 90 degrees, it is obvious that the plants were not straight. These plants either had 2 or 3 segments. As seen in this image, the plant on the left has a horizontal root. Once the plant grew out of the soil, the plant stem bent upwards and became vertical. The plant on the right did the same, however, its root is longer. The root is horizontal, and then it bends downward vertically. These plants have much more curvatures than the control group. These plants follow the rules of gravitropism, in which the stem grows upward in order to gain more sunlight, and the roots grow downwards in order to gain more water and nutrients. The plants in the microgravity environment varied a lot in its stem and root curvatures. Some appear to be very similar to the control group, which grew under normal gravity conditions. This may be due to the fact that the motor often stopped working and would not always be spinning. Although, some plants had strange stem and root curvatures, such as the one on the left. This plant appeared to have multiple loops and continually bends throughout its stem and root structure. This plant greatly varies from the control group, which has no curves at all. This may be due to the microgravity environment or due to the fact that the plant was not fully matured. In all, the hypothesis for the first experimental group, or the tilted plants, was supported by the qualitative data as seen in the study. Gravitropism causes stems to bend upwards and roots to bend downwards, however the plant may be positioned. Even though the cup was sideways, the plants grew out of the soil and bent approximately 90 degrees to grow towards the sky where the sun is often found, known as negative gravitropism. Similarly, the roots bent approximately 90 degrees downwards to the force of gravity in order to gain more nutrients and water, known as positive gravitropism. However, the hypothesis was not supported for the microgravity experiment. The hypothesis once again was that the plants would grow out of the soil and remain straight, similar to the control group, since the plant would not have any reference for gravity. Thus, it will grow normally based on the position of the soil. Ultimately, some of the plants grew off the walls of the cup towards the center of the cup, which was clear of soil. This growth was due to the displacement of soil. It may also be possible that centrifugal force acted upon the soil and the plants, since gravity was absent. Overall, there were a plethora of errors that may have occurred in this experiment. First of all, the plants may have not been planted in the optimal conditions. There could have been a variety of confounding variables, including differences in soil, light, and water conditions. In addition, for more data, the plants must be grown for a greater duration of time. If given more time, it is likely that the plants will grow more. Thus, the stem and root curvatures will be much more defined. Another major error that may have occurred was the rotation of the clinostat, which may have occurred too quickly. Since our motor was homemade and required a drill, the system often malfunctioned and had to be reset a lot. Since there was no way to ensure a constant pace of rotation, the clinostat most likely spun too quick. In addition, there was long durations of time when the clinostat did not spin at all.
The hormone responsible for gravitropism is called IAA, or indole acetic acid, which, in the presence of gravity, goes towards the bottom of the cell. In roots, IAA inhibits cell elongation. Thus, the root grows slower than the shoot. The opposite occurs in shoots. When there is a high quantity of IAA on the lower side of the cell, the shoot will grow upwards. Based on the influence of gravity on these organelles, chemical messengers are released to indicate which direction for which the plants to grow. These structures and cell types responsible for gravitropism are called amyloplasts. They are known as statoliths inside of the amyloplasts. Amyloplasts are specialized plastids that contain starches. These starches settle downwards based on which direction gravity is pulling down. The amyloplasts can be found in the cells of the shoots and the root cap of the plant. When a plant is not upright, then the amyloplasts will travel towards the bottom of the cell. It only takes a few hours for the plant to respond to this drastic change. The shoot or root will then grow in a new vertical direction. This occurs because amyloplasts physically come into contact with the endoplasmic reticulum, which induces the release of calcium ions inside of the endoplasmic reticulum. The calcium signals for the movement of a hormone known as insul acetic acid, which inhibits cell elongation in roots. Based on the influence of gravity on these organelles, chemical messengers are released to help tell the plant which direction to grow. The podcast Smarty Plants and the experiment which was conducted have many similarities. Both study plants' abilities to respond to a particular stimuli. Smarty Plants chose to study wind, while the experiment conducted chose to study the effects of gravitropism on plant growth. Although both experiments show signs of learning behavior, I would not compare it to the level of consciousness that is experienced by most animals and even insects. While the experiments do show a retained response to a particular stimulus or a pairing of stimuli, it should not, at this stage, be compared to consciousness or real memory in the sense of human interpretation of that term. The experiment which was conducted had some similarities to From Tree to Shiny Tree. The connections which the podcast talked about took many years to make, as well as the presence of fungus and other roots were necessary. These factors need many years to become a prominent part of the tree's ecology. If we allowed our plants to mature and grow for many more years, then more accurate comparisons could be made. But until more accurate time periods are shown, no accurate comparison can be made between the experiment and the podcast.